Narcolepsy type 1 is a chronic disorder of hypersomnolence caused by loss of the orexin-producing neurons in the brain. In this study, we aimed to map the orexin receptor 2 pathways that improve wakefulness and cataplexy in people with narcolepsy type 1. By activating these receptors, orexin receptor 2 agonists can promote sustained wakefulness and reduce cataplexy. Full alertness is driven by several types of neurons in the brain, and one especially important group releases the orexin neuropeptides. Normally, the orexins promote long periods of wakefulness. They do this by activating orexin receptors 1 and 2 in key wake-promoting brain regions. However, in people with narcolepsy, there's a selective and severe loss of the orexin-producing neurons. Consequently, people with narcolepsy have great difficulty staying awake for long periods, especially under quiet circumstances. The orexin neuropeptides also regulate rapid eye movement or REM sleep. With the loss of orexin signaling in narcolepsy, people also develop brief episodes of muscle paralysis and weakness known as cataplexy. This is probably the paralysis typical of REM sleep that inappropriately intrudes into wakefulness. Researchers have developed small molecule medications that activate orexin receptor 2, and clinical trials are showing great improvements in wakefulness, cataplexy, and other symptoms. Orexin receptor 2 is found in many brain regions, but it was unknown just where these medicines work in the brain to improve wakefulness and reduce cataplexy. To define the key pathways, we developed a mouse with the diphtheria toxin receptor selectively expressed in the orexin neurons. Treatment with diphtheria toxin resulted in a rapid 99% loss of the orexin-producing neurons, resulting in severe narcolepsy symptoms, including sleepiness and cataplexy. Treatment with OX201, an experimental orexin receptor 2 agonist, improved wakefulness and nearly eliminated cataplexy. To determine where in the brain orexin agonists work, we crossed these narcoleptic mice with mice in which expression of the orexin receptor 2 was blocked. In mice with fully blocked expression, the orexin receptor 2 agonist did not improve narcolepsy symptoms. By injecting a viral vector coding for Cre recombinase, we induced expression of orexin receptor 2 in the tuberomammillary region of the posterior hypothalamus. Now, the drug promoted long periods of wakefulness, but did not reduce cataplexy. Expressing orexin receptor 2 in the basal forebrain yielded the same results. We then restored orexin receptor 2 in the ventrolateral part of the periaqueductal gray in the brainstem. Now, the drug reduced cataplexy, but the ability of the mice to stay awake was only a little better. Overall, these experiments show that an experimental orexin receptor 2 agonist can promote wakefulness by activating neurons in the posterior hypothalamus and basal forebrain. But reductions in cataplexy are via brainstem neurons in the periaqueductal gray. These experiments highlight specific brain regions through which an orexin agonist can improve wakefulness and reduce cataplexy, but additional brain regions may also contribute. With this improved understanding of how orexin agonists work in the brain, our hope is that this will lead to better therapies for people with narcolepsy. Thank you.